That's not Giving Day. This is Giving Tuesday. It's a Giving Day, but not the Giving Day. You get it. But we're here up in the 4th floor of BTI at the Harrison Lab, and we're going to be guided around by one of the Harrison Lab members, Penelope Lindsay. Hello, Penelope.
Hi.
How you doing today?
Good. How are you?
Thank you for stepping away from your research to join us for a few minutes. So give us a little bit of an overview. I guess first tell us a bit about yourself, how you ended up coming to BTI and what your interests are.
Yeah, so I'm a 4th year PhD student in the Harrison Lab, and I've always been really interested in plant biology broadly and biotic interactions. So the Harrison Lab was a great fit for me because we study symbiosis, which is a beneficial plant, root, and fungus interaction. And so the plant gives the fungus carbon, and then in exchange for carbon, the fungus gives the plant phosphorus. And
which is good.
This is really good. Yeah,
Right.
Phosphorus is an important nutrient that all organisms need to survive. It's really important for DNA.
Oh,
and other
got a cart coming down the hallway.
Macromolecules. And it's extremely important for agriculture because there's a lot of fertilizers that use this phosphorus or need a lot of phosphorus in the fertilizers for the plants to be able to grow well. This excessive use of fertilizer use can be a bad thing though, because excessive fertilizer use can lead to runoff into water sources and cause
Right.
a lot of pollution.
I've seen pictures where lakes turn a very unnatural color.
There'll be really green algae blooms that can sometimes occupy entire lakes, even some of our Great Lakes. There's a really stunning image of Lake Erie, I believe, where there's this really intense green algae bloom that's indirectly as a result of this fertilizer runoff. And in addition, phosphorus is a finite resource. So there are all of these mines that are in Florida, for example, and other places around the world where you have just phosphorus being dug out of the ground, and it causes a lot of damage to the environment potentially. So it's really important that we find ways to more efficiently use the phosphorus that we have, and so symbiosis is one way that we can use it.
Phosphorus, it's kind of a finite resource, right?
Yep, it's a finite resource.
It just doesn't just keep being made.
No, so we need to be able to use it as efficiently as possible. So that's one of the ultimate goals of the Harrison Lab. And so we study symbiosis because of its beauty and also because it has potential agricultural benefits.
Absolutely.
Yeah.
All right.
So,
Well, what did you want to show us today?
okay, well, yeah, I'm going to take a tour of the lab, see what people are up to, and then I can also show you guys a transformed root that we use to visualize our AM association. So
Great. All right.
So let's take a tour.
Let's check it out.
All right, so we'll go ahead and see what everyone's up to. Yeah, this is one of several labs that we have here. This is Lena, who's a postdoc in the Harrison Lab. Actually, a high school. Yes. Yeah, I don't know if you guys want to introduce yourselves briefly and say what you're doing. But.
Yeah, I'm Lena. I'm a postdoc in the Harrison Lab. I'm here for a bit more than a year. I'm working on arbusculum mycorrhiza symbiosis, particularly trying to find out how maybe nutrient signaling influences mycorrhizal symbiosis. And this is Sophia. She's my high school intern. She's working here 12 hours a week.
Wow. Where do you go to high school?
Ithaca High School.
Ithaca High School. Okay. Well, we could have let your parents know. You could have tuned in.
Yeah, I only told her today.
It's for the best.
Yeah. Exactly. Yeah, we're doing DNA extraction at the moment.
Okay.
Lycopodium plants.
Yeah. So this lab has broadly referred to itself as the monocot lab because we work on a few different plant species in our lab, both dicots and monocots. We work with a model monocot called Brachypodium distachyon, and I focus mostly on Medicago truncatula, which is a dicot. And we can use so many different plant species because the AM symbiosis is formed between almost all land plant species, with some notable exceptions, such as probably the most well-known model plant, Arabidopsis. So it cannot form AM symbiosis. So focus on some of these other species.
I think we saw some of the plants you just mentioned down in the growth chambers this morning.
Yeah,
So we're bringing it full circle for you, for the viewers out there.
Yeah, so in this lab too, we do some protein work, and so no one's actively working with this right now, but I am often doing protein work to isolate my favorite protein, which is called vaporin, and I really like studying vaporin because it's an essential protein for the symbiotic association. So if you have plants that don't have vaporin, then the symbiosis can't proceed normally. So I'm trying to figure out why that is. And so I look at protein and cell biology experiments as well, and some genetics experiments, of course, because that's sort of the fundamental underpinning of a lot of what we're doing here. But yeah, so we have...
What's moving right here?
So this is our shaker, and it looks like somebody has maybe some root samples on here shaking, because when we're washing or rinsing roots, it's important for the buffer to evenly shake and sort of redistribute the chemicals that are coming out of the roots and stuff. So yeah, so it's
fun.
Yeah, this is our apparatus for setting electrical charges into our protein gels. These can get up to 250 volts, so it can be quite strong.
Yeah.
So definitely have to make sure everything's well protected for these.
It's good to know these are here. My winter's coming. My car might die in the parking lot. I'll come up here and get a jump. Beautiful.
Perfect. Yeah. You just have to find a way to get the car. Yeah, and so these are some of our rinse apparatuses here. Let's see. We've got additional incubators scattered around the lab. Let's move on. Oh, so Deirdre is not working right now, but she is our expert in doing different types of tissue transformations and has been creating a series of stable transgenic lines, which have different genes that, or proteins that will have some kind of fluorescent marker attached to them, so then we can look at them in the roots. And they'll glow with either a GFP or an M-cherry marker, and these will glow either green or red under the microscope, so then we're able to more easily, able to visualize them.
And do you have to utilize the imaging center for a lot of that work?
Yeah, we do quite a bit. Yeah, we're often using the confocal to be able to look at the subcellular localization of proteins at a single cell level, which is really great. Yeah, but we'll also use the stereoscope with a little bit. So here's Deirdre here.
There she is. She thought we were gone, I think.
I think I came right.
Yeah, would you mind telling me a little bit about what you're up to?
Sure. So I think I heard Penelope talking about the stable transformation process, but what I'm doing, I have sort of the end product where we usually infiltrate the agar that has the gene construct in it into callus, which these are two different species, but it will look sort of like a clump of undifferentiated cells, and as the process goes along, they will differentiate into roots and stems and whatever other plant parts. So this is the little metacargo plant. As you can see, it's got little leaves growing and roots growing at the bottom. So I'm about to transfer it to a different media, which will promote root growth. And then when it gets a little bit stronger, we can transfer it into soil. And this is the other species that I'm working on, which is Brachypodium. And it's a grass species, so it's a little more grassy. But you can see the little plant starting to form in the callus.
And if someone's just tuning in, they're wondering, so why are you working with all these little samples again? The answer would be kind of back to what you started off with, kind of the end game of...
Yeah, so we want to study these beneficial plant-fungus interactions to more fully understand the genetic mechanism that's underlying them. So then we can potentially use this to more efficiently harness phosphorus from the soil. So this has an ultimate agricultural benefit.
Absolutely. Great.
So this is where we do our agarose gel electrophoresis. So this is where we look at DNA. We have Sergey, who's looks like he's doing something with plates.
Yeah.
We could see what he's up to. You want to say what you're up to right now?
Yeah, what I'm doing now, I'm plating the agrobacterium on a plate, which later, in 2 days, I will use for transformation of medicago roots. And this agrobacterium, they contain special DNA molecules or constructs, which will be transformed into the medicago roots. And these constructs will allow us to sense phosphorus in the roots. We can check how phosphorus is flowing from the outside into the roots and where it's distributed in the cells in the roots. Yeah.
Great. Thank you, Sergey.
Yeah. Yeah, so we've got a really great number of postdocs, and a couple of PhD students who are in the lab. And we all have our own projects, but we all work together quite well, and everyone has a different kind of expertise. So Sergey is really great at something called cloning, where you're taking specific DNA molecules of interest and then putting them into your plants to express them, something like a transgenic line that has GFP, which was what I was just talking about, so you can look at your protein of interest in the plant. And he's also very good at microscopy. This is actually his poster. I don't know if this is of interest, but here are some of the images that we have. This is a single cell that contains an arbuscule, and there's a marker here that's marking the membrane that surrounds the arbuscule, which is the site of nutrient exchange between the plant and the fungus. And so it's really amazing that we're able to look at these structures. I think, yeah, but you can also have transgenic molecules that are marking the nuclei, which you can see are these circles that are in the cell. And pretty much any other subcellular component you can think of. So now I guess I can go ahead and show you a sample of how we are actually looking at these different transgenic molecules.
And a shout out to our supporters from the National Science Foundation and the Triad Foundation. Two of our partners that make a lot of this possible, so thank you to them.
Yeah, thank you. Okay, so this is my lab area. Just this bench is my bench here. So I have all of my different chemicals that I use for my different molecular biology processes. Also here I have a mature medicago plant. And this is a plant that's ready to be looked at under the microscope. So we grow them in these cones because it allows the roots to grow to a sufficient length to be colonized by the fungus. And we put a layer of sand, and this sand layer contains spores. And then after about a week or two, then the spores will germinate and begin to grow toward the root and then eventually colonize the plant. So the fungus is pretty much microscopic. You have these things that are called hyphae, which are kind of like the fungus root in a way. They're these very thin cylindrical structures that form. But you can't see them unless you're looking at them under the microscope. So here, I'm going to take out the plant, just squeeze it out of this cone. I mean, this is a very old plant, so it's a bit more difficult to get out, of course. But
things get stubborn as they get older.
They do.
I'm living proof of that.
So we have now this root system that's quite extensive at this point. And we're going to try to take as much of the soil out with our hands. It's quite a process here.
I love it. Live demo, though. This is great. Good TV.
And so then after we've pretty sufficiently taken the soil out, we'll rinse the rest of it in water, and this will get the rest out. There's the soil at the bottom here does not want to come out, but that's okay. We can cheat a little bit.
We won't tell.
cut the bottom of the roots off. And we often do this anyway because generally the bottom of the roots will not be colonized with the fungus. Okay, so now we have our roots, and we normally have 2 plants per cone. And we're going to distribute them evenly in this petri plate here. And now we have a root system that's ready to be visualized under our stereo microscope. So we can bring it into our stereo microscope.
This is where the magic happens.
Yeah, our lab definitely focuses a lot on cell biology. We really learn a lot from it.
I'm guessing we have to keep it dark in here?
Yeah, we have to keep it dark, and this is so we're able to sufficiently look at the fluorescent molecules, because otherwise it can be very difficult to visualize them unless they're really blaringly bright. So let's see here. Sometimes we'll turn on this lamp.
There's a little bit of light. That helps.
Lighting.
That's right.
Okay, so we just put the plant directly under the microscope here. And now we're ready to see whether or not it has its GFP signal. And in the case of this plant,
Is that the fungus? The signal?
So the signal is generally, we use plant proteins.
Okay.
And that's because it's a lot easier to transform a plant versus the fungus. The fungus, yeah, because of the nature of the way that the fungus cell biology is, it has a lot of cells that are all stuck together, pretty much. There are no discrete cells, so it makes it very difficult to transform and create fluorescent versions of the fungus. So we circumnavigate this by working with the plant. And so this particular plant has, I believe, a nuclear signal. So the nuclei should be glowing with GFP. So we can take a peek and see if we see something. So this light is our UV light, and it's got a filter for GFP specifically. And so this is going to be activating or allowing the GFP molecule to fluoresce, so we can actually look at it. So I can see here if there's any signal. Yep, and I see a signal here. So it's a bit difficult. I guess you can't really see what's going on through the microscope here.
We can take your word for it.
Yeah, but if you believe me, yeah, I can turn it on the big screen.
We're going to try to get it up on the screen.
Yeah, try to pull it up here. Takes a little while.
Penelope did all this hard work for us. We want to. We know how scientists like their results to be proven.
It's true. Yeah, so you can, the stereoscope is great because you can look at fluorescence, but you can also look at bright field images. So if you want to see whether or not your plants are colonized, for example, you'll have like a preponderance of hyphae, which, as I mentioned before, are the external or are the fungal structures that are kind of analogous to plant roots.
Well, I think it's just really shows how much kind of nuance there is in just doing. I mean, it sounds like your research is very focused, but then all the things that go into that focus as well is really something that needs to be appreciated. Yeah, no, everything is quite focused within the realms of our projects. We're very interested in the specific mechanisms underlying the development of our bug. Yeah, maybe we're gonna see here.
I can set it up with you.
Yeah, that sounds great. Thank you. So, yeah, so Armando is another postdoc in our lab, and Armando actually recently had a really great paper published in Nature Plants, and this, well, do you want to describe your paper? Or?
Well, we identified genes that are involved in mycorrhizal symbiosis, and now that we have all these genomes of plants that have been sequenced, we compare these genomes. And by a simple comparison, we identify the genes that are just specific for plants that form this interaction between plants and fungi.
Yeah.
That was basically it.
Well, congrats.
Yeah, no, it was great. And he also recently presented his research at ASPB, which is one of the biggest plant conferences that exists. Yeah, so that was in Austin back in July, and I happened to be there too. I presented a poster, but Armando presented a 20-minute talk about his research.
How often do you venture out of the Institute?
Right. So for conferences, generally once a year or so, each of us will go to one of them. We'll either go to one of the bigger ones like ASPB. MPMI was last year as well, and this conference is also a really big conference, but it focuses more specifically on biotic interactions. So either pathogen interactions with plants or some beneficial interactions with plants as well. But there are so many different scientific conferences out there, and it's a really great opportunity to network with people and gain additional insight into, different processes. It's really exciting. I feel like, when I went to ASBB, I learned so much, and so, yeah.
Yeah, it seems the science community is very collaborative in general.
Definitely, yeah, and that's one of the strengths that I really love about science, is that everyone really is working together, because when you work together, that's when you really make the greatest discoveries, I think. So, yeah.
So here,
Yeah. So
All right, we made it. Here's what's under the microscope.
Let's see if I can focus this a little better. So it's at a pretty high magnification right now. We're at 50x. And so it can be a little bit difficult to achieve really high resolution at this magnification. But you can see here, all of these dots are nuclear signal. You're going to have this nuclear signal in a specific layer of cells in the root too, and this is the cortical cells, which are sort of not right in the center of the root, but they're flanking the center of the root, because in the middle you have the vascular tissue where water and nutrients are transported throughout the plant. But the cortical cell layer is where the arbuscules actually form in the symbiosis. So we're often looking at cortical cells in our research. And so that's another reason why this signal is a little bit fainter, because in order to see it very clearly, you need to cut the root in half. And that's what we do when we're doing confocal microscopy. And this is what allows us to actually look at an individual cell. So,
Right. Well, Penelope, we have to wrap up so we can get down to our friends in the Mueller lab. But thank you very much for joining us and for opening up the Harrison lab to everyone remotely. We will be back in 4 minutes as we jump down to the Mueller lab to learn about bioinformatics. Do you ever work with the Mueller lab in any of that?
so there are some collaborations. In
my
fact.
work was a
Yeah.
collaboration with
Okay, great. Well, there you have it. See, collaboration right on cue. So thanks for joining us again on this Giving Tuesday as we go live from BTI throughout the day. And we'll be back in a good 4 minutes.
Yeah, thanks so much.